Hello and welcome to the first PowerPoint here for Unit 4 Area of Study 1. We're actually going to kick things off with an introductory PowerPoint just to review some of the key concepts when it comes to this topic. So we're calling this our sort of honorary KK0 topic. Like I said, it's really about providing background on some of the terms and concepts that we're going to explore in the subsequent notes for Area Study 1 of Unit 4. So we're going to start right back when and look at how, first of all, how we had a historical view around disease and now what the modern view is. So historically, if you've ever read books set in a period even far back as the Victorian era, obviously going back further as well, you might sometimes hear people talk about miasma. Historically speaking, there was this miasma theory that infectious diseases were caused by foul-smelling vapors in the air. Basically, miasma means bad air, and so stinky air was how disease was transmitted. Now, fast forward to the 1800s, and several people were working in this space, and two key individuals were Louis Pasteur and Robert Koch. And they were really fundamental in changing our way of thinking around disease from this miasma theory to germ theory, which is the modern view around disease. And germ theory is this idea that there are specific microbes that actually cause specific infectious diseases. And so we tend to call those microbes pathogens, and that's a term you've probably heard before. Louis Pasteur is remembered because he was able to show that it was microbes causing wine to go off and to start degrading and getting bad. And he actually was the one who just developed the process of pasteurization, which is the process we use with milk to make sure it is essentially fit to be stored and able to be transmitted long distances without going off and having people get sick from it. Robert Koch is really remembered as the founder of what we would call modern bacteriology, this understanding of not just microbes but specifically bacteria. And his focus was on anthrax, and he was able to show that anthrax was actually caused by Bacillus anthracis. And he also worked on other diseases, again being able to show that they were caused by microbes. But we particularly remember him from his work with anthrax. So when I say microbes, what do I mean? Well, those are organisms that are too small to see them without using a microscope. That's what the micro part means. They're tiny little things. And microbes are all around us. And just because I say microbes doesn't mean something bad. Microbes do include bacteria, fungi, and protists, but most microbes are totally harmless. They're actually completely fine. Some microbes can cause disease, and we call those ones pathogens. And sometimes certain forms of a microbe might be pathogenic, whilst other forms are not. And so pathogen is a term we use to refer to a disease-causing agent. Any microbe that can cause disease can be considered a pathogen. Pathogens have a lot of different forms. We generally group these into two main categories, our cellular versus our acellular. Unsurprisingly, the difference here is cellular ones are made of cells and the acellular ones are not. So cellular pathogens include parasites, protozoa, fungi, and prokaryotes. So parasites could be like tapeworm, for instance, as you can see there. There's a whole range of different ones. You could even consider lice as a form of parasite. Protozoa are single-celled organisms, but they're quite complex compared to bacteria. And this would include the protozoa that causes malaria, that's able to infect blood cells and cause that disease. Fungi, like athlete's foot, is one example. Ringworm, surprising thing, I know it says worm in the name, but ringworm is actually a fungal infection. That's another example of a cellular pathogen. And obviously we have the classic bacteria, and bacteria cause a whole range of things. You've got leprosy there as one example of a disease caused by a prokaryote, but even gastro that you might get is caused by E. coli. And so that's another example of disease that's caused by a cellular pathogen. Your acellular pathogens are your viruses and your prions. Viruses we're pretty familiar with, but just generally speaking, what they are is their genetic material surrounded by a protein capsule. And so a virus might have DNA inside of it, or it can even have RNA inside of it, and then it has some protein capsule around the outside to protect that genetic information and to help the virus inject that genetic information into a cell. On the screen, we've got a picture of a virus. That's actually a bacteriophage. That's a type of virus that actually infects bacteria, not necessarily human cells. One example of a virus would be the HIV virus, which causes AIDS, the disease. But we're generally familiar with plenty of viruses because the common cold is an example of a virus as well, and that's something we generally experience during winter too. Prions are an interesting category here of acellular pathogens. They're actually misfolded proteins. CJD, as you can see there, stands for 𝗘𝘅R𝗺 disease, and it's essentially the human equivalent of what we might call mad cow disease that we've seen in cows. It's an example of a prion disease where you have these proteins that might normally be present in the organism, but they get misfolded. And the way they get misfolded leads to them actually being quite harmful if they aggregate together. Particularly, we tend to find them aggregating in the brain tissue, causing damage to the brain tissue, which might lead an individual to appear mad, hence why we talk about mad cow disease. But again, this is essentially, it's a protein, and so it is not made up of a cell. It is just amino acids joined together and then folded in a way they shouldn't be. So these are our two categories, our cellular and our non-cellular pathogens, and we've got the examples there of each of them. So take a moment now, we've got an exam question here. It says cellular agents capable of causing infection of body cells include A, prions, B, toxins, C, DNA viruses, or D, gram-negative bacteria. You can take a moment, pause the video, answer the question, and then you can go through the answer in the video linked in the description. Alrighty, so how do we know if something's a pathogen? How do our bodies recognize them? This leads me to another word that you need to be familiar with, and that's the word antigen. So pathogens are, as we've talked about, either made up of cells, they might be a single cell or they might be multicellular, or they're one of these acellular examples like a virus or a prion. But at the end of the day, all of these pathogens have some elements of the molecules on their surface that are able to be recognized by elements of our immune system. Some of them are more easily recognized than others. Others tend to fly under the radar a bit, and that can pose a real problem for us in dealing with those. But generally speaking, anything that's on the surface of the pathogen that allows it to be recognized and generates a response within the immune system, that molecule is called an antigen. And we'll look in detail about how antigens actually get recognized by our immune cells, but just know for now that when we are talking about a pathogen, we are referring to either the whole cell or the whole cellular organism, or it's a virus or a prion. When we talk about antigen, we are talking about a molecule that pathogen has that is able to be recognized and is able to generate a response from the immune system. So that's what an antigen is. Generally speaking, the types of molecules that are antigens are proteins, glycoproteins, or glycolipids. The glyco there means it's got like a carbohydrate chain, so a protein is just a protein by itself. A glycoprotein is protein plus a carbohydrate chain. And glycolipid is a lipid, so like a fat or an oil, with a carbohydrate chain also attached. Now, there are some scenarios where instead of being on the surface of the pathogen, the antigen is actually a free-floating molecule that triggers the immune system, and that's the sort of thing we see with snake venom and how that triggers an immune response. Different pathogens have different antigens. Now, if pathogens are related, for example, smallpox and cowpox, the similar type of virus, we find that they might have similar antigens, and sometimes they're similar enough that our bodies can recognize antigens on pathogens it's never been exposed to, but it might have been exposed to the similar one. So for instance, being exposed to cowpox was discovered historically to provide immunity to smallpox because the pathogens had very similar antigens, and by being able to recognize the ones for cowpox, it could also recognize, our bodies could also recognize the ones for smallpox. This isn't super common. It's not across all pathogens, but it certainly can happen. But generally speaking, we'd say different pathogens have different antigens. And so over time, your immune system's learning to recognize all those different pathogens based on the antigens. And as it recognizes them and remembers them, it can respond quicker and quicker, which is really helpful for us because it hopefully means future exposures do not necessarily result in us getting sick. Speaking of getting sick, one place we notice people getting a little confused with words is between infection and disease. And so infection and disease actually mean two different things. An infection just means that the pathogen got into the body and has begun to multiply. Disease, on the other hand, is when the cells are actually damaged as a result of that infection, and you start seeing signs and symptoms. So what this means is that an infection can occur, but it may or may not lead to the development of the disease. If you have the disease present, it must be caused by an infection. There must be an infection present. But just because you have an infection does not mean it will progress to the disease. So you might be exposed to the flu virus and you might have some of it enter your body. But if your body is able to recognize and get rid of that nice and quickly, that infection may never develop into you having the flu. So there's a connection between these two terms, but one does not necessarily lead to the other. And you can see some examples of pathogens down the bottom there, which cause diseases in the body if they get to that point where they're able to cause that damage, but not necessarily will they get to that disease point. Here we have another exam question, and it says, it's reasonable to infer that an infection has occurred if A, histamine is released by cells, B, a scab has formed on a cut in the skin, C, bacteria are found in the large intestine, or D, pathogens are found in leg muscle tissue. So you can pause the video now, have a bit of a think, put down your answer, and then go check it against the video linked in the description. So how do we get from infection to disease? Well, for starters, your pathogen has to get into the body somehow. So it's got to overcome whatever barriers there are. It has to get to the cells that it affects. Now, for some pathogens, it might affect any cell in the body. Others might be more specialized in terms of they might only have an impact on certain cells. So they're having to overcome the body's defense systems, not just in terms of barriers, but all the other things circulating within the body in our other lines of defense. That pathogen has to become established at one site. It doesn't have to just get there. It has to start multiplying and really get settled in that space. And it needs to multiply rapidly to overcome those defense systems as well. From there, it actually has to then cause damage and harm to the host, leading to the symptoms of the disease being produced. So those are all the steps that we'd expect a pathogen to have to go through to actually result in disease. Now, sometimes this is really quick, other times it takes a little bit longer. And that's where we're talking about incubation periods. You can be exposed to the pathogen and become infected, sometimes in only a short period of time before you start exhibiting symptoms, sometimes a significantly longer period of time before you exhibit symptoms. So the incubation period is the time between when are you infected versus when do the first symptoms appear. During that time frame, the pathogen's multiplying. And so those can be very, very short because the pathogen's really quick, or it can be really, really long because it might take the pathogen a really long time. You will also notice that if you look on the diagram there, it starts off down the bottom having a latent period, and then it moves into a period of communicability. And that period of communicability is a fancy way of saying you're contagious. Unsurprisingly, when you're exhibiting the disease, you are clearly contagious. You can see the arrows at the top that say infectious. It means the same thing here. But you'll notice that you can be infectious or you can be contagious during the last bit of the incubation period, which might mean that you are infectious or contagious prior to exhibiting symptoms, which the longer that period where there's no symptoms but you're infectious, the longer that overlap is, the more you can spread the disease. And you can see in our table on the right that we have quite a difference in incubation periods for different diseases. Influenza and the common cold are very, very short incubation periods. You're generally exposed, then a day to two later, you're going to exhibit symptoms. This is why you might hang out with someone who's got a cold, and then the next day or 2 days later, you also have a cold. Really, really short incubation period. If you look down the list, you'll see diseases like measles and smallpox have much longer incubation periods. And with measles, you might also find that there is a decent period in there in which somebody is infectious, but not showing symptoms yet. And that's a real problem with diseases like measles, because it means people go about doing their everyday lives whilst being infectious and spreading the disease about. And so measles can, when somebody's got it, spread quite easily. It's often why we see with measles, if we think someone's gone to, say, a sporting event with measles, it actually gets quite a bit of attention because we don't want these to have a big outbreak, but they can, because you can actually affect quite a number of people in that time frame. If you scroll right down to the bottom of the list, you'll notice that those are really big incubation periods. And in fact, AIDS, which is caused by the HIV virus, has a 1 to 10 year incubation period. So somebody can be infected with AIDS or infected with the HIV virus, which would lead to AIDS potentially, but not know, and even be highly infectious for many years, which, depending on someone's lifestyle, might mean they're actually able to infect quite a large number of people with that, which is one of the reasons in certain communities we see it spread really far and wide. So there's a range of different incubation periods for different diseases. How do you know if someone's sick? Well, generally they have symptoms. And so symptomatic is a term we use to refer to someone who is infected and they have the disease and they're showing all the signs and symptoms of that disease. If someone is symptomatic, there's a good chance that they are currently infectious. It is possible for, at certain points of a disease, for different diseases, that they might show some symptoms but not really be in an infectious period anymore. Though generally better to be safe than sorry, and you generally want to just keep a little bit of distance between you and the person who is showing symptoms. On the other hand, some people can be asymptomatic. They're infected, they have the disease, but they're not showing signs and symptoms of that disease. This is a problem because they can also, in this time frame, be infectious. So you've got somebody who has no symptoms but can actually spread the disease. This is essentially when we talk about someone being like a typhoid Mary and spreading a disease. Well, typhoid Mary was a real lady who was able to spread typhoid because she was carrying the disease, or she's carrying the pathogen, but she wasn't affected and showed symptoms like other people were. And so she was able to spread the disease far and wide. With diseases, it's worth noting that you could be talking about infectious diseases or non-infectious diseases. Because our topic here is immunology and we're looking at the immune system, we're really going to just deal with infectious diseases. These are ones that are transmissible or communicable, and so you can pass them on from one person to another. These ones are caused by specific infectious agents, our pathogens, or they are caused by toxic products pathogens produce. And so that pathogen can be transmitted from an infected person, it could be transferred from an animal, or even what we call a reservoir to a susceptible host, somebody or something that can become infected by that pathogen. Non-infectious diseases, on the other hand, are not things that can spread from person to person. So this could be something caused by environmental conditions. It could be something like scurvy, which is due to a vitamin C deficiency. That's a disease, but it's not infectious. It doesn't get transmitted from person to person. It gets caused by not having enough vitamin C in your diet. And this also covers inherited diseases, say haemophilia, which is a genetic disorder. And so these are non-infectious diseases. They're not generally tied to the immune system. And so we're not going to focus particularly on these. Most of our focus will be on the immune system dealing with infectious diseases. Some of the basic ways diseases actually get transmitted, or pathogens get transmitted, are through one of these 4 categories: direct contact, contamination, airborne, and vectors. And you can break these down further, but these generally cover most, if not all, modes of transmission. So direct contact is physical contact between individuals where one's infected and they pass it on to someone else. It may even be through contact with objects that have gotten touched by someone who is unwell. It also includes the transmission of diseases via exchange of bodily fluids. And so that might be in somebody's got a cold, so their saliva has the pathogen in it and they kiss their partner and they're able to infect them that way, for instance. Contamination is really around food and drink. And so, for instance, if you have an infected water source, say it has the pathogen for dysentery in it, and you're drinking that water is contaminated, and that is how that pathogen can get passed on to someone else, giving them dysentery. It also might be food that's got an excess of E. coli because it's been left out too long, and then you get some gastro or something like that from it. or just a bit of food poisoning. Airborne diseases are able to be transmitted via coughing and sneezing, though if they can only be transmitted within a short distance, they're more a direct form of direct contact. Airborne do require a longer distance to be able to be covered by them and still be viable to infect people. Finally, vectors. Vectors are where there is some other organism between individuals. So you have one person who's infected, one person who's not, and it doesn't go directly from the person who's infected to the non-infected person. It goes through some other organism, like an animal or an insect first, and then gets passed on to the non-infected person. And so this would be something like malaria, where malaria gets passed on because you have a person who has malaria, gets bitten by a mosquito. The mosquito sucks up their blood, which contains the malarial pathogen. And then that mosquito buzzes off and it goes to suck somebody else's blood, transmitting back the malarial pathogen to this new person. And so in that case, the mosquito is acting as the vector. So that is our main ways that we see disease being transmitted. Here is our last question. It says Chagas disease, a potentially life-threatening illness in humans, is caused by the protozoan parasite 𝕃𝕚𝕟𝕚𝕚. The disease is found mainly in Latin American countries where the parasite is spread by an insect called a 𝕃𝕚𝕠𝕚. In this example, the role of the 𝕃𝕚𝕚 is to A. Introduce the virus into the protozoan parasite, B. Infect the insect with Chagas disease, C. Introduce the gene into a bacterium, or D. Enable 𝕃𝘅R𝗺 to enter the host. So you can take a moment, pause the video, have a think, write your answer down, and then you can go and check the answer in the video linked in the description. So that brings us to the end of the PowerPoint. If there's anything you didn't understand, you can go back and rewatch that section or reach out to your teacher for further clarification. Otherwise, I'll see you in the next video. Bye.